There's something profoundly humbling about standing at the edge of a mystery so deep, even modern science struggles to explain it. Picture this. Inside every living cell, there are machines tiny, intricate, and impossibly precise, working in perfect harmony to sustain life. But here's the catch. These machines are so complex, so finely tuned, that the idea they emerged purely by random chance feels almost like believing a hurricane could assemble a jumbo jet from scattered parts. This isn't just a thought experiment, it's the reality of biology, where countless mechanisms defy the straightforward narratives we've been taught about evolution. Consider the hummingbird, a creature so agile it seems to defy physics, its wings beating with such speed and precision that engineers can barely replicate it. Or the human eye, a masterpiece of optics so sophisticated that even Charles Darwin admitted the thought of it evolving step by step made him shudder. These aren't just quirks of nature, they're puzzles that whisper a tantalizing question. What if life's most astonishing designs aren't accidents at all? What if they're clues pointing to something far more profound, something science is only beginning to grasp? That's where our journey begins. Today, we're exploring 22 biological wonders that stand as towering enigmas in the face of conventional evolutionary theory. From the eerie synchrony of symbiotic relationships where two organisms depend on each other so completely that neither could survive alone, to the mind-bending complexity of DNA, a code so elaborate it puts our most advanced software to shame. These aren't just gaps in our knowledge, they're chasms, forcing us to rethink how life truly came to be. So buckle up, because by the time we're done, you'll see the natural world in a whole new light, questioning everything you thought you knew about the origins of life itself. The bacterial flagellum is like a microscopic outboard motor, spinning at speeds that would make a Ferrari jealous, up to 100,000 revolutions per minute. But here's what really stops scientists in their tracks. This isn't just a simple propeller. It's a machine built from around 40 protein parts, each meticulously arranged like gears in a Swiss watch. Remove just one piece and the entire system grinds to a halt. No propulsion, no survival advantage, just a useless molecular relic. This is the heart of what biologists call irreducible complexity. Think of a mousetrap. If you're missing the spring or the platform, it doesn't just work poorly, it doesn't work at all. The flagellum poses the same dilemma. Evolutionary theory suggests complex systems arise through tiny, functional steps, each offering some benefit. But what's the halfway point for a flagellum? A partial rotor without a stator is like a car engine without pistons. It might as well not exist. Even Darwin acknowledged that if a structure couldn't be formed through numerous, successive, slight modifications, his theory would break down. Yet the plot thickens. Some researchers argue certain flagellum proteins share similarities with other bacterial systems, like the type 3 secretion system, a molecular syringe used to inject toxins. Could this be an evolutionary stepping stone? Maybe, but here's the catch. While the parts might overlap, the functions diverge wildly. Repurposing a syringe into a rotary motor is like turning a bicycle pump into a jet engine. The odds of random mutations not only preserving but repurposing these components with perfect precision stretch credibility. And then there's the assembly manual. Building a flagellum isn't just about having the right parts. It's about delivering them to the construction site in the correct order, a process guided by layers of genetic instruction. Mess up the timing and the proteins pile up like a junkyard instead of a motor. It's not just a machine, it's a masterclass in nanoengineering, So where does that leave us? The flagellum isn't just a challenge to evolution, it's a mirror held up to the limits of our understanding. Whether you see it as evidence of unguided tinkering or something more intentional, one thing's certain: this tiny marvel forces us to confront the awe-inspiring sophistication of life's smallest architects. The human eye doesn't just capture light, it performs a high-speed molecular ballet so precise, it makes the most advanced camera look like a child's toy. At the heart of this miracle is the phototransduction cascade, a process where photons of light trigger a domino effect of chemical reactions, converting the physical world into electrical signals your brain can understand. But here's the staggering part: this entire system operates at near-perfect efficiency, with each step perfectly timed down to the millisecond. Miss one link in the chain, and vision collapses into chaos. Take the humble rod cell, a light-sensitive neuron in your retina. When a single photon strikes a molecule called retinal, a component of rhodopsin, it triggers a shape change so precise it's like flipping a switch. That tiny shift sets off a protein relay race, activating hundreds of molecules in sequence, amplifying the signal like a microscopic avalanche. Within milliseconds, this cascade closes ion channels, hyperpolarizing the cell and sending a crisp signal to your brain. The entire process is so finely tuned that even slight mutations can lead to night blindness or total vision loss. Darwin himself wrestled with this. In On the Origin of Species, he called the idea of the eye evolving through natural selection absurd in the highest possible degree, yet he argued that given enough time, even the unimaginable might become plausible. But modern biochemistry has only deepened the mystery. The phototransduction cascade isn't just complex, it's irreducibly interdependent. Remove any one of its core proteins, transducin, phosphodiesterase, or cyclic GMP, and the system fails completely. There's no functional half-eye to bridge the evolutionary gap. Some scientists propose that simpler light-sensitive cells, like those in jellyfish or flatworms, could be evolutionary precursors. But here's the catch: those cells lack the molecular machinery for signal amplification or rapid resetting key features that make vertebrate vision so sharp. It's like comparing a candle to a laser. Even if early light detection systems existed, the leap to the vertebrate eye's sophistication would require not just new parts, but an entirely new assembly line. Random mutations tinkering with individual proteins couldn't orchestrate such a synchronized overhaul. And then there's the reset problem. After the cascade fires, every molecule must be meticulously recycled to prepare for the next photon. This cleanup involves at least 5 dedicated enzymes working in concert like a pit crew servicing a race car mid-lap. Without this reset, your vision would fog up after one blinding flash. The odds of all these mechanisms emerging simultaneously by chance, astronomically low. Yet here we are, seeing in vivid detail, thanks to a system so optimized it borders on the miraculous. Whether you view it as evolution's greatest triumph or a puzzle begging for deeper answers, one thing's clear: the phototransduction cascade doesn't just challenge our assumptions, it illuminates the breathtaking precision hidden in every glance. Imagine a tiny, self-contained city where power plants, libraries, and transportation systems all work in perfect harmony, except this city is a cell, and its complexity makes the most advanced human metropolis look primitive. This is the eukaryotic cell, nature's great leap forward from the simplicity of bacteria to the intricate architecture of life as we know it. At its core lies one of biology's most baffling puzzles. How did a humble prokaryote, a cell without a nucleus or organelles, transform into something so sophisticated that it became the building block of every plant, animal, and fungus on Earth? The leading explanation, Endosymbiotic Theory, suggests a dramatic plot twist. Around 2 billion years ago, a host cell supposedly swallowed a bacterium whole, but instead of digesting it, they struck a deal. The bacterium became the mitochondria, the cell's power generator, while the host provided protection and resources. It's a compelling story, but the details unravel under scrutiny. For starters, this wasn't just a casual partnership. It was a full-scale merger requiring dozens of synchronized innovations. The mitochondria had to surrender its independence, offloading critical genes to the host's nucleus while retaining just enough DNA to function. Meanwhile, the host cell had to develop entirely new systems to import mitochondrial proteins, coordinate their production, and ensure both entities divided in lockstep during reproduction. Think of it like two companies merging overnight, except one hands over its entire IT department while the other rewires its infrastructure to accommodate the change, all without a single glitch. The odds of this happening through random mutations are vanishingly slim. Even more perplexing is the timing. Mitochondria didn't just bring energy, they supercharged it, enabling cells to grow 100 times larger than their prokaryotic ancestors. But without pre-existing mechanisms to manage that energy surplus, the first eukaryotic cells would have imploded from metabolic chaos. It's like installing a nuclear reactor in a village that still uses candles without first inventing power grids or safety protocols. Then there's the question of the nucleus. Unlike mitochondria, its origins remain even murkier. Some scientists propose it evolved from folded membranes that trapped DNA, while others suggest viral infections might have played a role. But neither theory explains how the cell's genetic material became so meticulously organized, with histones wrapping DNA like spools and nuclear pores acting as bouncers, deciding which molecules enter or exit. These features aren't just add-ons, they're essential for survival. A cell without a functional nucleus is like a factory without a foreman. Materials pile up, blueprints go missing, and production grinds to a halt. And let's not forget the sheer improbability of two independent organisms merging their replication cycles. Mitochondria divide on their own schedule, yet somehow, eukaryotic cells evolved mechanisms to ensure they split in perfect sync with the host. Miss this coordination by even a fraction, and the cell either starves from lack of energy or drowns in toxic byproducts. So where does this leave us? The leap to eukaryotes isn't just a step in life's history, it's a quantum jump, defying the gradual, piecemeal process Darwin envisioned. Whether through a freakish sequence of luck or forces we've yet to understand, one thing's clear: this cellular revolution remains one of evolution's most astonishing unsolved mysteries. The genetic code isn't just a set of instructions, it's a masterwork of engineering so flawless. It puts the most sophisticated computer algorithms to shame. Every living organism, from the tiniest bacterium to the tallest redwood, relies on the same elegant system to translate DNA into proteins, the building blocks of life. But here's what's truly mind-bending. This code isn't just functional, it's optimized with built-in safeguards that minimize errors and resist corruption. If evolution were truly a blind, unguided process, why does this universal language look so meticulously fine-tuned? Take redundancy, for example. The genetic code is written in 3-letter sequences called codons, each corresponding to a specific amino acid. But here's the twist: there are 64 possible codons, way more than the 20 amino acids they encode. This means multiple codons often spell out the same amino acid, like synonyms in a sentence. At first glance, that seems wasteful, but dig deeper and you'll find a brilliant fail-safe. When mutations strike, they're most likely to alter the 3rd letter of a codon, the one that matters least. So even if a random change occurs, it often lands on a synonym, leaving the protein intact. It's like a typo in a word where the meaning stays clear no matter how you misspell it. Then there's error minimization. Some amino acids are chemically similar. Think of them as interchangeable parts in a machine. The code is structured so that mutations are most likely to swap one for another, causing minimal disruption. For instance, replacing leucine with isoleucine is like swapping two nearly identical screws. The machine keeps running, but if the code were arranged randomly, mutations would frequently substitute wildly different amino acids, scrambling proteins beyond repair. The odds of this precise arrangement emerging by chance, roughly 1 in a million. Even more baffling is the code's universality. If evolution were constantly tinkering, we'd expect to find wildly different versions across species, each optimized for its own niche. Yet with rare exceptions, every organism shares the same core blueprint. This conservation suggests something startling. The code may have hit a peak of efficiency early on, leaving little room for improvement. But how natural selection works step by step, yet the code's perfection seems to demand a leap, a system where all the pieces had to align just right from the start. Some theorists argue the code evolved through a series of historical accidents, frozen in place once life became too complex to change it. But that raises another puzzle. Why hasn't evolution stumbled upon even marginally better versions in billions of years of trial and error? Others propose the code's optimization points to deeper physical or chemical constraints, laws we haven't yet uncovered, but neither explanation fully accounts for its precision. What we're left with is a paradox. The genetic code is too elegant to be a fluke, too universal to be a frozen accident, and too resilient to be the product of unguided tinkering. Whether you see it as the signature of something deeper or simply evolution's greatest stroke of luck, one thing's certain: this isn't just a language of life. It's a cipher waiting to be decoded. Bacteria don't just fight viruses, they wage molecular warfare with a precision that would make special forces envious. At the heart of this defense is CRISPR-Cas9, a system so advanced it's been dubbed genetic scissors. But here's what leaves scientists stunned: it doesn't just cut randomly. It remembers. When a virus attacks, bacteria store snippets of the invader's DNA in a library called CRISPR, clustered regularly interspaced short palindromic repeats. If the same virus strikes again, the bacteria retrieves that genetic mugshot, crafts a guided RNA missile, and unleashes Cas9 to slice the enemy's DNA to ribbons. It's not just immunity, it's a molecular intelligence agency operating with chilling efficiency. The sheer elegance of this system defies easy explanation. For CRISPR to work, multiple components must align with near-perfect coordination. First, the bacteria must recognize and capture viral DNA, then integrate it into its own genome without disrupting essential genes. Next, it transcribes this memory into RNA and pairs it with Cas9, a protein that acts like a scalpel guided by GPS. The precision is staggering. Cas9 doesn't just cut anywhere. It navigates the massive expanse of the bacterial genome to find one exact sequence, a feat like finding a single sentence in a library of encyclopedias blindfolded. Evolutionary theorists face a dilemma. For natural selection to favor CRISPR, each step capture, integration, transcription, and targeting would need to provide an immediate survival advantage. But individually, these pieces are useless. Storing viral DNA without the ability to use it is like keeping a stack of wanted posters with no police force. And Cas9, without its RNA guide, is just a protein in search of a purpose. The system only works when all parts exist simultaneously, fully formed. Some argue simpler precursors might have existed, perhaps standalone enzymes that cut DNA randomly, offering partial protection. But randomness is the opposite of what CRISPR achieves. A blind knife can't become a precision tool through incremental tweaks. It requires a complete overhaul of its targeting mechanism. The leap from haphazard cutting to guided gene editing isn't just a step, it's a paradigm shift. Then there's the question of memory. How does a bacterium know to archive viral DNA, then deploy it later? This isn't passive adaptation, it's active strategy, a form of molecular foresight that challenges our definitions of intelligence. Even more perplexing is CRISPR ubiquity. Nearly half of all bacteria and most archaea have some version of it. If this system evolved once, it's a fluke, but repeated across countless species, that hints at a deeper logic woven into life's fabric. What makes CRISPR truly astonishing isn't just its existence, it's that we've harnessed it to edit our own genes. The same mechanism bacteria used to fend off viruses now lets scientists rewrite the code of life, curing genetic diseases and reshaping ecosystems. But here's the irony: we needed intelligence to repurpose this tool. So how did blind, unguided processes engineer it in the first place? The answer, like CRISPR itself, remains tantalizingly out of reach. Butterfly metamorphosis isn't just transformation. It's a biological magic trick so radical, it rewrites the rules of what we thought possible. Picture a caterpillar, a creature built for one purpose, eating. It munches leaves, grows exponentially, then one day it stops. What happens next defies logic. The caterpillar essentially dissolves itself, turning its own body into a soupy puddle of cells. From this chaotic broth, an entirely new organism emerges, a butterfly with wings, compound eyes, and a proboscis, none of which existed in the caterpillar's original blueprint. This isn't just change, it's a total reinvention, as if a bulldozer melted down into a helicopter mid-construction. The sheer audacity of this process raises a glaring question. How could such a dramatic overhaul evolve step by step? Metamorphosis isn't a slight tweak. It's two completely different body plans encoded in one genome with a self-destruct button in between. For Darwinian gradualism, this poses a paradox. Intermediate stages would be disastrous. Imagine a half-caterpillar, half-butterfly struggling to survive, too clumsy to crawl, too malformed to fly. Natural selection favors functional advantages, yet every transitional form here would be a liability. Then there's the matter of genetic foresight. The caterpillar's cells carry instructions not just for its current form, but for an entirely different organism it will become. Key genes lie dormant, activated only when the time is right, like a script with two acts written in advance. Some of these genes even trigger the caterpillar's digestive enzymes to essentially eat itself alive, recycling its own tissues into raw material for the butterfly. This level of coordination suggests something beyond random mutation. It's as if the genome knew the end goal before the journey began. Scientists point to ancient hormonal switches like ecdysone and juvenile hormone, which regulate the timing of metamorphosis. But these signals are just the conductors. The orchestra is the DNA itself, playing a symphony of transformations with no room for error. Miscalculate the hormone levels, and you get a stuck pupa, neither caterpillar nor butterfly, just a biological dead end. The precision required here is mind-boggling, especially when you consider that butterflies and moths alone account for over 150,000 species, each with variations on this theme. Some theorists suggest metamorphosis evolved from simpler molting processes, where arthropods shed their exoskeletons to grow. But molting is like trading in an old suit for a larger one, not dismantling the wearer and rebuilding them from scratch. The leap to complete metamorphosis would require not just new genes, but an entirely new developmental playbook, where cells must forget their original identity and reprogram en masse. Random mutations tinkering with molting hormones couldn't orchestrate such a radical rewrite. And then there's the energy problem. Metamorphosis is a metabolic marathon requiring stockpiled reserves to fuel the rebuild. Caterpillars are basically eating machines for this very reason. But how did the first proto-butterflies know to binge in advance? Without pre-existing adaptations for storing and redirecting energy, the process would collapse mid-transformation. So what are we left with? Metamorphosis isn't just a challenge to evolutionary theory. It's a window into life's capacity for reinvention. Whether you see it as nature's greatest improvisation or evidence of something deeper, one thing's certain: this isn't just change. It's alchemy. Sexual reproduction shouldn't work. Think about it. Asexual organisms clone themselves effortlessly, doubling their numbers with each generation, while sexual species invest massive energy in finding mates, competing for partners, and shuffling genes. A process so inefficient, it leaves half the population males unable to produce offspring at all. From a cold Darwinian perspective, sex is a losing strategy. Yet here we are in a world dominated by creatures that rely on it, from fruit flies to blue whales. This isn't just a curiosity, it's a paradox that cuts to the heart of evolutionary theory. The textbook explanation is genetic diversity. By mixing DNA, sex creates variation, helping populations resist parasites and adapt to changing environments. But here's the catch: that benefit only matters after sexual reproduction is already up and running. Early stages of this transition would have been disastrous. Imagine a primitive organism trying to switch from cloning to sex. First, it must evolve mechanisms to produce sperm and egg cells that carry half the usual DNA. Then it needs complementary partners with compatible reproductive systems. Until both pieces are in place, sex is a dead end. Worse, those first partial steps would reduce fitness, since half the offspring inherit only a fraction of their parents' successful genes. It's like rewiring a car engine while it's still running, except the car is racing for survival. Some scientists propose that sex began as a repair mechanism. Early cells might have fused temporarily to fix damaged DNA, swapping genes like spare parts. Over time, this occasional teamwork could have become mandatory. But this raises another puzzle. Repair doesn't explain why organisms would abandon the simplicity of cloning for the complexity of meiosis, the intricate cell division that produces sperm and eggs. Meiosis isn't just mitosis with a twist. It's a molecular ballet involving precise DNA cutting, recombination, and chromosome pairing. A single misstep can lead to sterility or genetic disorders. How could such a fragile system evolve piecemeal when every intermediate stage offers no obvious advantage? Then there's the Red Queen hypothesis, named after Alice in Wonderland's character who runs constantly just to stay in place. The idea is that sex keeps species one step ahead of pathogens, which evolve rapidly to exploit static hosts. But again, this only works if sex is already established. Predation can't explain its origin, only its persistence. The most startling clue comes from ancient fossils. Evidence suggests sex emerged around 1.2 billion years ago, coinciding with the rise of eukaryotic cells. Did the complexity of these cells demand a new reproductive strategy, or was sex somehow entangled with the very origin of multicellularity? The answers remain locked in Earth's deepest rocks and in the DNA of every sexually reproducing organism alive today. One thing's certain: this isn't just a quirk of biology. It's a revolution that reshaped life itself, leaving us with a mystery as profound as the act of creation. The blood-clotting cascade is like a molecular tightrope walk. One misstep, and the result is either catastrophic bleeding or a deadly clot. This system relies on over 20 proteins interacting with split-second precision, each activating the next in a chain reaction so finely tuned that even minor defects can lead to disorders like hemophilia or thrombosis. But here's the puzzle. How could such an intricate, interdependent network evolve gradually when every step along the way demands perfect coordination just to function at all? Take thrombin, the central player in this drama. It's a protein that does double duty, slicing fibrinogen to create sticky fibrin threads that form clots, while also regulating its own production to prevent runaway clotting. Thrombin doesn't work alone, though. It's activated by another protein, which is itself activated by yet another, tracing back through a branching pathway of enzymes and cofactors. Remove any single link, say factor 8 or factor 9, and the cascade collapses. A person with hemophilia lacks just one of these factors, and even a minor cut can become life-threatening. Evolutionary theory faces a steep challenge here. For clotting to evolve, each protein would need to provide some survival advantage on its own. But individually, most of these factors are useless. Factor 10 doesn't help unless factors 5 and 2 are already in place, like trying to start a car with only the ignition switch and no engine. Intermediate stages would either clot uncontrollably, killing the organism, or fail to clot at all, leading to fatal bleeding. Neither scenario offers a survivable stepping stone. Some researchers point to simpler clotting systems in invertebrates, like horseshoe crabs, as possible precursors. Their blood relies on a basic 2-protein cascade to trap bacteria in gel-like clots, but the leap from this rudimentary defense to the vertebrate system is enormous. It's not just about adding more proteins. It's about integrating them into a self-regulating feedback loop where each component modulates the others. Imagine upgrading a bicycle into a jetliner mid-flight. Every addition would need to synchronize perfectly with systems that didn't yet exist. Then there's the question of timing. Clotting must strike a balance between speed and control. Too slow and an animal bleeds out. Too fast and clots choke off blood flow. Vertebrates achieve this through inhibitors like antithrombin and protein C, which put the brakes on the cascade at just the right moment. These safeguards aren't optional extras, they're essential. Without them, the first clot would spiral into a fatal thrombosis. Yet, from an evolutionary standpoint, these inhibitors only make sense after the clotting proteins they regulate are already in place. So what's the solution? Some scientists propose gene duplication as a possible escape hatch, accidental copying of existing genes, allowing one copy to mutate into a new function. But even if a duplicated gene stumbled into a proto-clotting role, it would still need to interface seamlessly with the rest of the system. The odds of this happening repeatedly across dozens of proteins stretch plausibility. The blood clotting cascade doesn't just challenge evolutionary theory, it exposes a paradox at the heart of complexity itself. Systems this finely balanced seem to demand either foresight or freakish luck. Whether you see it as a testament to nature's ingenuity or a riddle still waiting for an answer, one thing's clear: every scab on your skin is a miniature masterpiece of biological engineering. The human brain doesn't just process information, it conjures symphonies, wrestles with existential questions, and even ponders its own existence. This isn't merely computation, it's consciousness, the most profound mystery in biology. How can 3 pounds of gelatinous tissue firing electrical signals give rise to the vivid sensation of a sunset, the sting of regret, or the thrill of discovery? Evolutionary theory stumbles here not for lack of trying, but because the gap between neurons and experience is more than a step, it's a chasm. Consider the so-called hard problem of consciousness, coined by philosopher David Chalmers. Even if we map every synapse and decode every neurotransmitter, we're left with a glaring disconnect. Why does any of this feel like anything at all? A computer can simulate weather patterns without getting wet, yet your brain doesn't just simulate pain, it hurts. This qualitative leap from physical processes to subjective experience defies conventional explanations. Natural selection favors survival traits, but what survival advantage comes from the ache of nostalgia or the awe of starlight? Some scientists argue consciousness is an emergent property, a byproduct of complexity, like wetness arising from countless water molecules. But this analogy falls short. Wetness can be measured, consciousness remains stubbornly intangible. Others propose integrated information theory, suggesting that any system with sufficient interconnectedness might harbor some form of awareness. Yet even this framework doesn't explain why interconnected neurons should produce a unified sense of self. A pile of sand has countless interactions between grains, but no one suggests it's contemplating its own granularity. Then there's the evolutionary timeline. Basic awareness, say, a fish avoiding predators, might offer clear survival benefits, but how does that scale to human self-reflection? At what point did neurons begin not just reacting to stimuli, but wondering about them? The leap from instinct to introspection lacks fossil evidence or plausible intermediates. You can't half-experience a thought. Even more perplexing is creativity. The brain doesn't just solve problems, it imagines possibilities that don't exist, from fictional worlds to mathematical abstractions. These aren't survival tools, they're leaps into the void. Picasso's Guernica or Einstein's theory of relativity didn't help them evade predators, yet the same biological machinery that keeps hearts beating produced these masterpieces. Neuroscience has uncovered astonishing details: mirror neurons, default mode networks, synaptic plasticity. But these are mechanisms, not explanations. They describe how the brain works, not why it feels. Until we bridge that gap, consciousness remains evolution's most tantalizing enigma, not just how life survives, but how it means. The relationship between figs and wasps isn't just cooperation, it's a biological lock and key system so precise, it borders on the surreal. Picture a fig tree. Its fruit isn't actually a fruit, but a hidden garden of inverted flowers, sealed inside a fleshy pod. To pollinate them, the tree doesn't rely on wind or bees. Instead, it has a dedicated partner, a tiny wasp, often no bigger than a grain of sand, evolved to navigate this labyrinth with eerie precision. But here's the catch: the wasp can't survive without the fig, and the fig can't reproduce without the wasp. Their fates are so tightly bound that one's existence is the other's lifeline, a dance of dependency that defies the odds of random chance. Take the female wasp's journey. She burrows into the fig through a pore so narrow, it often tears her wings off a one-way trip. Inside, she lays her eggs in some of the flowers, while pollinating others with pollen carried from her birth fig. Then she dies, her body absorbed by the fig as fertilizer. When her offspring hatch, the males emerge first, wingless and blind, with one mission: mate with their sisters, then chew an escape tunnel for them. The females, now dusted with fresh pollen, fly off to repeat the cycle in another fig. Miss a single step, the timing, the tunnel, the pollen transfer, and the entire system collapses. This isn't just symbiosis, it's a biological Rube Goldberg machine. For the partnership to evolve, both species needed to develop complementary traits simultaneously. The fig had to grow the exact right pore size for the wasp, produce flowers perfectly sized for her eggs, and time its fruit production to match her lifespan. Meanwhile, the wasp needed specialized organs to collect and deposit pollen, instincts to navigate the fig's interior, and a life cycle synced to the tree's reproductive rhythm. Half-formed adaptations would be useless. A wasp that couldn't pollinate would starve the fig. A fig without the right pore would trap the wasp. Evolutionary theorists face a dilemma. Gradual step-by-step development can't easily explain this mutualism because the intermediate stages offer no survival benefit. A proto-wasp that occasionally wandered into figs wouldn't gain an advantage unless the tree already had traits to exploit her. Likewise, a fig with a partial pore or poorly timed flowering would waste resources attracting the wrong visitors. The odds of both species stumbling into perfect compatibility through random mutations are astronomically low, like two locksmiths independently inventing matching keys. Some scientists argue the relationship began with generalist wasps pollinating open flowers, then gradually specializing. But figs have been tightly linked to their wasps for over 60 million years, with over 700 fig species, each paired to its own wasp. That level of specificity suggests not just adaptation, but co-engineering. Even more baffling is the wasp's sacrifice. Evolution favors traits that enhance individual survival, yet these wasps die to serve the fig's reproduction, a paradox that challenges selfish gene theory. What makes this partnership truly astonishing isn't just its complexity, it's its fragility. A single invasive species or climate shift can unravel centuries of coevolution. Yet here it stands, a testament to life's capacity for intricate collaboration. Whether you see it as a statistical fluke or something more intentional, one thing's clear: the fig and the wasp don't just coexist. They're two halves of a single story, written in the language of survival itself. Enzymes are nature's ultimate chemists, performing feats that leave even the most advanced laboratories in awe. These molecular machines accelerate chemical reactions by factors of millions, sometimes trillions, with such precision that they can pluck a single target molecule from a cellular soup of thousands. Picture a bustling train station where one security guard instantly spots a single suspicious passenger among millions, escorting them directly to a waiting train while ignoring everyone else. That's enzyme specificity, a level of discrimination that seems almost supernatural when you consider these proteins emerged from the chaos of random mutations. The secret lies in their active sites, the enzyme's reaction hub. These pockets aren't just generic hollows, they're exquisitely shaped to fit their target molecules called substrates, like gloves molded to individual hands. This lock and key model, first proposed over a century ago, barely captures the sophistication at play. Modern research reveals active sites as dynamic structures, often reshaping slightly to grip their substrates even tighter, a process called induced fit. The enzyme hexokinase, for example, clamps down on glucose like a Venus flytrap, enclosing it completely to prevent unwanted side reactions. The precision is staggering. A single misplaced atom in the active site can render the enzyme useless, like a key bent ever so slightly that it no longer turns in its lock. This perfection poses a puzzle for evolutionary theory. For an enzyme to evolve, each intermediate stage must offer some advantage. But incomplete active sites are worse than non-functional, they're often dangerous. A partially formed enzyme might bind the wrong molecules, triggering toxic side reactions or depleting essential compounds. It's not just about building a better catalyst, it's about avoiding catastrophic interference with the cell's delicate chemistry. Consider the classic example of bacterial beta-lactamase, an enzyme that breaks down penicillin. This protein doesn't just resist the antibiotic, it dismantles it with surgical precision, snipping a specific bond in the penicillin molecule while ignoring nearly identical structures. Evolutionary biologists suggest this ability emerged from mutations in pre-existing enzymes. But here's the catch: the intermediate forms wouldn't have provided incremental resistance. They'd have been like a broken scissors, useless until fully formed. Without the complete active site geometry, early beta-lactamases couldn't weaken penicillin enough to matter. The bacteria would still die, making each mutation along the way evolutionarily invisible. Then there's the question of folding. Enzymes are chains of amino acids that must twist into perfect three-dimensional shapes to function. Misfolded proteins aren't just inactive, they can clump together, causing diseases like Alzheimer's. Yet the odds of a random amino acid sequence folding into a functional enzyme are vanishingly small. Calculations suggest it would take longer than the age of the universe for chance alone to produce even one simple enzyme. Some researchers propose that early enzymes were far sloppier, catalyzing multiple reactions weekly before specializing. But this raises another riddle. Modern cells can't tolerate such ambiguity. Metabolism depends on enzymes discriminating ruthlessly between nearly identical molecules. A single error, say, confusing citrulline for ornithine in the urea cycle, can be fatal. Evolution would need to explain not just the origin of enzymes, but their abrupt transition from generalists to specialists without disrupting existing pathways. Perhaps the most mind-bending examples are catalytic RNAs or ribozymes, which suggest a world before protein enzymes existed. These RNA molecules can cut and splice themselves, hinting at a primordial RNA world. But ribozymes are orders of magnitude less efficient than protein enzymes. The leap from crude RNA catalysts to today's precision tools would require not just new materials, but an entirely new engineering paradigm. What we're left with is a paradox. Enzymes are too perfect to be accidents, too essential to have emerged gradually, and too intricate to have been invented whole. Whether you view them as evolutionary masterstrokes or evidence of deeper principles at work, one thing's certain: life runs on molecular machinery whose origins remain as mysterious as its mechanisms are flawless. The monarch butterfly's migration is one of nature's most astonishing feats, a journey so precise, it borders on the miraculous. Every year, millions of these delicate insects travel up to 3,000 miles, navigating from as far north as Canada to a handful of specific mountain peaks in central Mexico. But here's what defies logic. The butterflies that arrive have never been there before. They're separated by generations from their ancestors who last made the trip. Somehow, without maps or mentors, they inherit an itinerary written into their DNA, a celestial roadmap combining sunlight, magnetic fields, and an internal clock. This isn't just instinct, it's a biological GPS so advanced, it challenges everything we thought we knew about inherited behavior. Start with the sun compass. Monarchs don't just fly south, they adjust their path based on the sun's position, compensating for its movement across the sky throughout the day. This requires not just light sensitivity, but an internal clock, a circadian rhythm synchronized with the solar cycle. Experiments show that if you disrupt their circadian rhythms by altering light exposure, the butterflies lose their directional sense entirely. It's like trying to navigate with a broken watch and a spinning compass. Then there's the magnetic component. Monarchs possess cryptochromes, light-sensitive proteins in their antennae that detect Earth's magnetic field. This acts as a backup system on cloudy days, ensuring they stay on course even when the sun vanishes. But here's the catch: these proteins didn't evolve in isolation. They had to integrate seamlessly with the sun compass, creating a fail-safe navigation network. A monarch with just one system would be like a pilot relying solely on landmarks in fog, doomed the moment conditions change. The real mystery, though, lies in the generational handoff. The butterflies that leave Mexico in spring only live a few weeks, breeding along the way. Their descendants continue north, then produce the super generation, that makes the return trip months later. How does a lifespan measured in weeks encode a migration spanning seasons? This isn't just memory, it's a time-delayed relay race where the baton is passed through DNA. Evolutionary biologists struggle to explain how such a system could emerge piecemeal. Partial navigation offers no survival benefit. A monarch that flies randomly southward until it hits Mexico wouldn't leave more offspring than one that stayed put. The entire system, sun compass, magnetic sensors, circadian clock, had to function flawlessly from the start. Even more perplexing is the precision of the destination. The wintering sites are so specific that deforestation of just a few acres can devastate entire populations. How could random mutations guide butterflies to these tiny oases across continents? Some suggest monarchs originally followed shifting food sources, gradually refining their routes, but this ignores the genetic programming required. A butterfly doesn't learn the route, it's hardwired at birth. Others propose ancestral populations were once continuous, with migration emerging as climates changed. Yet fossil evidence shows monarchs have navigated long distances for millions of years, long before recent ice ages. Perhaps the most haunting question isn't how they do it, but why. Migration is perilous. Predators, storms, and exhaustion claim countless lives. What evolutionary pressure could justify such risk? The answer may lie in the wintering sites themselves. The cool mountain microclimates slow the monarch's metabolism, allowing them to survive months without food. But this just pushes the mystery back a step. How did they know these sanctuaries existed before finding them? The monarch's journey isn't just a biological process, it's a poem written in the language of genes, a testament to life's capacity for wonder. Whether you see it as evolution's greatest puzzle or a whisper of deeper patterns, one thing's certain: these fragile wings carry not just a species, but a mystery that defies simple explanation. Imagine a world where every screw, every bolt, every gear in every machine had to twist in exactly the same direction, not roughly, not mostly, but perfectly, universally identical. That's the reality inside your cells right now. The molecules of life, amino acids in proteins, sugars in DNA, exist in two mirror image forms like left and right hands. Chemists call this chirality. In the wild, non-living chemistry produces equal amounts of both versions. But here's the kicker: life ruthlessly enforces a single-handed monopoly. Every protein in your body uses only left-handed amino acids. Every strand of DNA contains only right-handed sugars. This isn't just a preference, it's a universal law of biology, and its origins might be the most overlooked enigma in the story of life's beginnings. The problem starts with assembly. Molecules with mixed chirality don't fit together. Picture trying to build a house where half the nails have reverse threading, the structure collapses. For life's molecular machines to function, every piece must lock into place with geometric perfection. Enzymes, those precision catalysts we marveled at earlier, have active sites shaped to accept only one chiral form. Insert the wrong version, and the enzyme stalls like a key jammed in a lock. Even more critical is DNA's double helix. Its iconic spiral depends entirely on right-handed sugars. Flip just one sugar molecule, and the helix unravels, taking genetic information with it. So how did nature solve this universal jigsaw puzzle? Random chemical reactions can't explain it. When scientists simulate early Earth conditions sparking amino acids from simple gases like Stanley Miller's famous experiment, they always get a 50-50 mix of left and right-handed forms. Yet somehow life bypassed this symmetry, committing exclusively to one version. Some theories suggest subtle physical forces might have tipped the scales. Circularly polarized light from neutron stars, for instance, can slightly favor one chiral form when irradiating organic molecules. Earth's mineral surfaces, like quartz crystals, also exhibit chirality, potentially concentrating one form through adsorption. But these effects are vanishingly small, like a coin landing on edge once in a billion tosses hardly enough to explain life's absolute uniformity. Then there's the snowball hypothesis. Once a slight imbalance existed, perhaps in a primordial pool of molecules, self-replicating systems could have amplified that tiny bias exponentially. Think of a snowball rolling downhill, gathering mass, except here, the mass is molecular purity. The problem, this just pushes the mystery back a step. Even a 1% chiral preference would require a mechanism to prevent the wrong molecules from contaminating the system during replication. Modern cells have elaborate proofreading enzymes to maintain homochirality, but early chemistry had no such quality control. The most startling clue comes from meteorites. The Murchison meteorite, which fell in Australia in 1969, contained amino acids with a slight excess of left-handed forms. If extraterrestrial delivery seeded Earth's chemistry, could space itself be inherently chiral? Maybe, but space is also brutally random. Cosmic rays and thermal chaos would likely scramble any delicate imbalances over millions of years. What makes homochirality truly baffling isn't just its existence, it's its universality. From the deepest ocean vents to the highest stratosphere, every organism we've ever discovered adheres to the same handedness. This suggests the choice wasn't just frozen by accident, it was foundational, wired into life's operating system from the very first spark. Whether that spark carried a hidden asymmetry we've yet to discover, or whether some deeper principle guided the selection, remains one of biology's most profound unsolved puzzles. One thing's certain, the molecules inside you aren't just building blocks, they're relics of a decision made billions of years ago, a choice that shaped all life since. The immune system operates with the precision of a master locksmith, crafting keys for locks it has never seen. At the heart of this defense are B cells, tiny factories that generate antibodies, custom-built proteins designed to latch onto invading pathogens with unerring accuracy. What's staggering isn't just their precision, but their versatility. Your body can produce billions of distinct antibodies, each tailored to recognize a unique molecular signature. This astronomical diversity arises from a genetic lottery so chaotic it defies belief, yet so finely tuned it borders on artistry. The process begins in your bone marrow, where B cells shuffle, cut, and paste segments of DNA like a molecular deck of cards. 3 gene families, V, D, and J, recombine randomly, creating unique sequences that code for antibody variants. But randomness alone wouldn't suffice. Enter hypermutation. Once a B cell encounters a threat, it deliberately mutates its antibody genes at a rate 1,000,000 times faster than normal, refining the fit like a blacksmith hammering a molten blade. The result, antibodies that cling to viruses or bacteria with near-perfect specificity. Here's the paradox: this system appears to anticipate pathogens before they're even encountered. How could evolution design a mechanism to combat threats that don't yet exist? Traditional Darwinian models struggle here. Natural selection favors traits that solve immediate problems, yet antibody diversity is a solution in search of future problems, a biological crystal ball. Some scientists argue the system isn't truly prescient. It's a shotgun blast of randomness where a few lucky B cells inevitably hit their marks, but this overlooks the elegance of the feedback loop. Successful antibodies trigger clonal expansion, flooding the body with protective copies while useless variants are discarded. It's not just trial and error, it's trial, refinement, and archival. Your immune system remembers past invaders, ensuring faster responses upon repeat exposure. This immunological memory is why vaccines work and why you don't suffer endlessly from the same cold. Yet questions linger. The recombination process is mediated by enzymes like RAG, which slice DNA at specific spots to enable shuffling. These enzymes didn't evolve for this purpose. They're thought to have originated from ancient viral proteins co-opted into the genome. That's like finding a scalpel in a cave and discovering it perfectly fits modern surgery. Even more perplexing is the system's restraint. Hypermutation could easily spawn cancerous mutations or self-attacking antibodies, yet fail-safes like central tolerance weed out dangerous variants before they wreak havoc. The immune system doesn't just defend, it learns. It's a living library of potential threats, written in a language of genes and proteins. Whether you see it as evolution's greatest improvisation or evidence of something deeper, one thing's undeniable. This biological masterpiece blurs the line between randomness and design, leaving us to wonder how much of life's complexity is accident and how much is architecture. Plants aren't supposed to move, at least not like this. The Venus flytrap defies every expectation we have about the pace of botanical life, snapping its jaw like leaves shut in a mere tenth of a second. That's faster than you can blink. What's even more astonishing is how it achieves this feat without a single muscle or nerve. This isn't just movement, it's a high-speed ambush, orchestrated through a blend of electrical impulses and hydraulic engineering that would make a robotics engineer envious. At the heart of this carnivorous marvel are trigger hairs, tiny sensory bristles lining the inner surface of each leaf. Touch one of these hairs twice within 20 seconds, and the trap initiates an action potential, an electrical signal remarkably similar to the nerve impulses firing in your own body. But here's where it gets wild. The flytrap doesn't just react, it counts. 2 touches are required to avoid wasting energy on false alarms like raindrops or wind-blown debris. This isn't reflex, it's calculation, a primitive form of decision-making in an organism without a brain. Once the signal fires, the trap's mechanics kick into overdrive. The leaves, held open by structural tension, suddenly release like a stretched rubber band snapping back. This happens through a rapid shift in water pressure within the plant cells. In their default state, the outer leaf layers are convex, bowed outward, but when triggered, they flip to concave in a fraction of a second, propelled by cells that lose their rigidity as water rushes between them. It's a hydraulic masterstroke, one that requires not just speed but perfect synchronization. A delay of milliseconds would let the prey escape. The evolutionary origins of this system are shrouded in mystery. Most plants move glacially slow, their growth measured in days or weeks. So how did the flytrap's ancestors stumble into such radical acceleration? One theory suggests it began with simple leaf curling, a common response to touch in many plants. Over time, mutations might have exaggerated this movement, favoring those that could trap insects for extra nutrients in nitrogen-poor soils. But the leap from sluggish curling to lightning-fast snapping demands more than incremental change. It requires a complete overhaul of cellular architecture, sensory signaling, and energy storage, all converging toward a single, ruthless purpose. Even more perplexing is the trap's selectivity. After closing, the plant assesses its catch. If the struggling prey continues stimulating the trigger hairs, the trap seals tightly and begins secreting digestive enzymes. If not, it reopens within hours, conserving energy. This implies not just movement but feedback, a level of responsiveness that blurs the boundary between plant and animal behavior. Then there's the question of energy. Rapid movement is costly, yet the flytrap thrives in nutrient-starved wetlands where other plants wither. Its solution: A metabolic gamble. Each snap consumes a significant portion of the plant's energy reserves. Miss too many strikes and it risks starvation. This delicate balance suggests an evolutionary trade-off so precise, it borders on the improbable. The Venus flytrap doesn't just challenge our definitions of intelligence and motion, it forces us to reconsider what life is capable of. In a world where plants are often dismissed as passive, this carnivorous prodigy stands as an electrifying reminder: nature's creativity knows no bounds. Whether you see it as evolution's improvisation or something more enigmatic, one thing's clear: the flytrap's snap isn't just a meal ticket, it's a declaration. Bats don't just navigate the darkness, they rewrite the rules of perception. While we fumble for light switches, these winged marvels paint detailed mental maps using nothing but sound, turning echoes into precision guidance systems. Their echolocation is biological engineering at its most extreme, a symphony of specialized anatomy, neural computation, and split-second timing that allows them to dodge obstacles and snatch insects mid-flight, all in pitch blackness. The sophistication of this system doesn't just stretch evolutionary theory, it snaps it like a twig under the weight of irreducible complexity. Consider the physics at play. A bat emits ultrasonic pulses, some reaching frequencies beyond 120 kHz, 5 times higher than human hearing can detect. These cries last mere milliseconds, yet the bat's brain measures the returning echoes down to microsecond precision. That's like distinguishing between two clicks separated by a millionth of a second, a temporal resolution that would make the most advanced stopwatch blush. This timing isn't just impressive, it's essential. A delay misjudged by even a few microseconds could mean the difference between catching a moth and smashing into a tree. But timing alone isn't enough. The bat's ears are masterpieces of acoustic engineering. Their tragus, a fleshy projection in the ear, acts like a radar dish, funneling returning echoes toward the eardrum. Some species can even adjust ear shape mid-flight to fine-tune sensitivity. Meanwhile, their brains perform real-time calculus, converting minuscule delays into spatial coordinates. The result, a 3D soundscape so vivid, bats can detect objects thinner than a human hair. Here's where evolution faces a paradox. For echolocation to work, all components must exist simultaneously. A bat with ultrasonic vocal cords but no specialized ears would deafen itself with its own cries. One with sensitive ears but no neural processing would drown in meaningless noise. Even the emission mechanism is a marvel. Bats produce pulses at pressures loud enough to damage human ears, yet their own hearing remains protected by muscles that briefly disconnect their eardrums during each shout. This isn't just adaptation, it's a biological Rube Goldberg machine where every gear must mesh perfectly from the start. Some scientists point to simpler precursors like shrews that use click-based navigation. But shrews lack the bat's precision. Their clicks are broadband and short-range, more akin to tapping a cane than orchestrating sonar. The leap to ultrasonic frequencies, directional beams, and neural echo mapping isn't incremental, it's a revolution. Even more baffling is the diversity of bat sonar. Over 1,000 species echolocate, each with unique call designs, from frequency-modulated sweeps to constant tones. Did this complexity evolve once, then radiate, or independently in separate lineages? Fossil evidence offers no clear answers, as soft tissues like vocal cords rarely fossilize. Then there's the brain power. A bat's auditory cortex is hypertrophied, dedicating vast neural real estate to echo processing. These aren't passive reflexes, they're active computations, integrating Doppler shifts to track moving prey. Some bats even adjust call patterns to avoid jamming each other's signals, a form of acoustic etiquette. This level of sophistication implies not just hardware, but software, innate algorithms fine-tuned over eons. So how did blind chance assemble such a system? Random mutations tinkering with vocal pitch might produce ultrasonic cries, but without paired ear adaptations, they're useless. Natural selection can't favor a half-built sone any more than a plane can glide with one wing. The bat's echolocation isn't just a tool, it's a manifesto written in ultrasound, declaring that some biological innovations defy piecemeal explanations. Whether you hear in it the hum of evolutionary ingenuity or the echo of deeper mysteries, one thing's certain: in the darkness, bats have found a light that science is still struggling to see. The firefly's glow isn't just a pretty spectacle, it's a biochemical marvel that puts our most advanced lighting technology to shame. Deep in the insect's abdomen, a reaction unfolds with such precision, it borders on alchemy. Luciferin, a light-emitting molecule, meets its match in the enzyme luciferase, with oxygen serving as the spark. Together, they produce light with 100% efficiency, converting every ounce of energy into pure illumination without wasting a single photon as heat. Compare that to the humble light bulb, which squanders 90% of its energy as warmth, or even LEDs, which still bleed off 20%. The firefly's lantern is a masterclass in energy economy, a feat of natural engineering that science has yet to replicate in the lab. What makes this glow even more astonishing is its control. Fireflies don't just light up randomly, they flicker with intention, using specific flash patterns to communicate, attract mates, or warn predators. Each species has its own Morse code, a rhythmic sequence of pulses that's as distinctive as a fingerprint. The male Photinus carolinus, for example, synchronizes its flashes with thousands of others in a dazzling light show, turning entire forests into living constellations. This isn't just chemistry, it's a language written in photons, demanding not just the right ingredients but the neural circuitry to orchestrate them. The evolutionary origins of this system are shrouded in mystery. Bioluminescence exists elsewhere in nature: deep-sea fish, fungi, even bacteria, but the firefly's version is uniquely efficient. Most theories suggest it began as a detox mechanism, with luciferase originally breaking down harmful oxygen radicals. But detoxification doesn't explain the leap to light production. A primitive enzyme neutralizing toxins wouldn't benefit from emitting photons. It's like a car evolving a radio before inventing wheels. For natural selection to favor bioluminescence, each intermediate step would need to offer a survival advantage. Yet a faint, inefficient glow in an ancestral beetle would hardly deter predators or attract mates. The system only becomes useful once it's bright and controlled, a classic chicken and egg dilemma. Then there's the question of coordination. The firefly's light organs are packed with specialized cells called photocytes, layered like lenses to amplify and direct the glow. These cells are wired directly to the insect's nervous system, allowing millisecond-level precision in flash duration and timing. The biochemistry is just one piece. The entire organism must be tuned to exploit it. A firefly with luciferin but no neural control would be like a lighthouse stuck on visible but useless for signaling. Perhaps the most haunting detail is the reaction's purity. Human chemists have spent decades trying to replicate cold light, yet our best efforts still fall short. Even the luciferase enzyme itself is a paradox. It accelerates the reaction without being consumed, like a match that never burns out. Such perfection hints at a deeper optimization, as if life stumbled upon a universal principle of energy conversion and refined it to its absolute limit. Whether you see the firefly's glow as evolution's finest accident or a whisper of nature's hidden blueprints, one thing's undeniable: In a world where energy is precious, this tiny insect has perfected the art of turning chemistry into magic. Its light doesn't just pierce the darkness, it illuminates the boundaries of what biology can achieve. The Arctic tern doesn't just migrate, it redefines the limits of endurance and precision. Every year, this featherweight navigator embarks on a staggering round-trip journey from the Arctic to the Antarctic and back, covering over 50,000 miles, the equivalent of flying twice around the Earth. What's truly mind-bending isn't just the distance, but the accuracy. These birds return to the same nesting grounds with pinpoint consistency, correcting for wind drift, storms, and even subtle shifts in celestial cues. Their flight paths aren't rough estimations, they're masterclasses in biological triangulation, suggesting an internal GPS so advanced it rivals modern satellite systems. At the heart of this feat lies a suite of navigational tools that would leave even seasoned pilots in awe. Arctic terns calibrate their routes using the sun's position, maintaining a mental time-compensated compass that adjusts for its movement across the sky. By night, they likely read the stars, their brains wired to recognize constellations' rotational patterns like ancient mariners. But celestial navigation isn't foolproof. Clouds obscure, and stars pivot with the seasons. So the terns layer in more cues, Earth's magnetic field, detected through cryptochrome proteins in their eyes, and even olfactory landmarks, with some evidence suggesting they smell their way across featureless oceans. This isn't just one system, it's a redundant, fail-safe network, where the failure of any single cue can be compensated by others. The evolutionary puzzle here isn't just the complexity, it's the irreducibility. For migration to offer a survival advantage, the entire toolkit must function from the start. A tern that could only follow the sun would veer off course at night or under clouds. One that only sensed magnetism might misread local anomalies. The system only works as an integrated whole, raising the question, how could such a multifaceted mechanism evolve piecemeal? Intermediate stages, partial navigation with intermittent success wouldn't justify the immense energy costs of such long-haul flights. A tern that got lost halfway wouldn't leave more offspring, it would leave none. Then there's the matter of calibration. Young terns on their first migration make the journey alone without guidance from experienced adults. Their navigational algorithms aren't learned, they're hardwired, fine-tuned by eons of selection. But how does instinct account for Earth's ever-changing geophysics? Magnetic poles drift, coastlines erode, and atmospheric conditions shift over millennia. Yet the tern's internal maps remain eerily accurate, as if programmed with an updatable database. Some scientists propose that shorter migrations preceded these epic voyages, with incremental expansions over generations. But this fails to explain the precision. Navigating from Greenland to Iceland is one thing. Hitting a speck of land in the Antarctic after months at sea is another. The difference isn't just degree, it's kind. Others suggest genetic drift or even hybridization could have played a role, but these are just labels for mystery, not mechanisms. Perhaps the most humbling detail is the tern's endurance. These birds spend nearly their entire lives in daylight, chasing endless summer across hemispheres. Their wings, spanning just 30 inches, withstand gales, salt spray, and exhaustion that would ground any aircraft. Every flap is a testament to optimization, from their aerodynamic feathers to their salt-excreting nostrils. The Arctic tern's journey isn't just a migration, it's a manifesto written in wing beats, declaring that life, against all odds, can conquer time and space. Whether you see it as evolution's boldest gamble or a navigational riddle still unsolved, one thing's certain: this bird doesn't just follow the horizon. It masters it. The octopus doesn't just blend in, it becomes its surroundings with a fluidity that defies biology. Imagine a creature that can shift from jagged coral to smooth sand in seconds, its body morphing in color, texture, and even three-dimensional shape to vanish before your eyes. This isn't mere camouflage, it's biological wizardry, orchestrated by a trio of specialized skin structures working in perfect harmony. Chromatophores, tiny sacs of pigment, expand or contract like microscopic balloons to create instant patterns. Beneath them, iridophores act like living prisms, reflecting light to produce metallic blues and greens. And then there are the papillae muscular bumps that rise and fall to mimic textures, from seaweed fronds to rocky crevices. Together, they form a dynamic disguise system so advanced, it makes Hollywood special effects look amateurish. What's truly baffling isn't just the mechanics, it's the intelligence behind them. An octopus doesn't passively react to its environment, it interprets it. In lab experiments, these cephalopods have matched complex backgrounds they've never encountered, suggesting an innate ability to deconstruct visual information and translate it into physical mimicry. A Caribbean reef octopus, for example, can replicate the checkered pattern of a chessboard or the stripes of a lionfish, despite having no prior exposure to either. This implies not just reactive adaptation, but a cognitive library of templates, a mental gallery of disguises waiting to be deployed. The evolutionary origins of this system are a minefield of paradoxes. For chromatophores to evolve, they'd need to offer immediate survival benefits, but early, rudimentary versions say patches of pigment that only partially expanded would hardly conceal the animal. In fact, they might make it more conspicuous, like a half-finished mural drawing attention to the wall. The system only works when all layers, color, reflectivity, texture are fully operational. An octopus with chromatophores but no papillae would be like a chameleon trying to hide on a corrugated roof. The colors might match, but the flat silhouette would give it away. Then there's the question of control. Each chromatophore is wired directly to the octopus's nervous system, allowing millisecond adjustments. But unlike our muscles, which contract in response to conscious thought, the octopus's camouflage operates largely unconsciously, as if its skin has a mind of its own. Some research suggests the animal's arms contain autonomous neural networks, meaning they can decide how to blend in without central brain input. This decentralization raises another riddle. How does natural selection favor a system where the body outthinks the brain? Perhaps the most haunting detail is the mimic octopus, a species that doesn't just hide, it impersonates. By contorting its body and adjusting its colors, it can mimic venomous sea snakes, flounders, or even lionfish, deterring predators by pretending to be something more dangerous. This isn't camouflage, it's theater, a performance requiring knowledge of other species' appearances and behaviors. The odds of such mimicry evolving through random mutations stretch plausibility, like a spy learning a foreign language by flipping through a dictionary at random. The octopus's vanishing act isn't just a survival tactic, it's a silent revolution, challenging our assumptions about intelligence, evolution, and the very nature of perception. Whether you see it as nature's greatest sleight of hand or a puzzle science has yet to solve, one thing's clear. In the ocean's shadowy depths, the line between seeing and believing dissolves like ink in water. Deep in the mangrove forests of Southeast Asia, a spectacle unfolds each summer night that seems to defy the very nature of randomness. Thousands of fireflies flashing in perfect unison, their tiny lights pulsing like a synchronized galactic heartbeat. This isn't just coincidence, it's a phenomenon so precise it borders on the supernatural. The synchronous fireflies of Malaysia and Thailand don't merely glow, they communicate in a silent, rhythmic language, their bioluminescent signals locked into a shared tempo without any central conductor or external cue. It's as if every beetle in the swarm shares an invisible metronome, their flashes rippling across the trees in waves of light so coordinated, early explorers mistook them for flickering lanterns hung by human hands. The secret lies in a delicate dance of biology and physics called emergent coordination. Each firefly's flash is governed by its own internal clock, but here's the twist: these clocks aren't isolated. When a firefly sees its neighbor's flash, its internal rhythm subtly adjusts, speeding up or slowing down to match the collective pulse. Over time, these tiny corrections snowball into perfect synchrony, transforming individual flickers into a luminous chorus. This isn't just mimicry, it's a form of biological democracy, where every participant votes with its light and the majority rules. The neural machinery behind this feat is staggering. Fireflies possess specialized photoreceptors tuned to detect the exact wavelength of their own species' glow, filtering out distractions like moonlight or rival species signals. Their brains then process this visual input through feedback loops that act like biological phase-locked loops, the same technology engineers used to synchronize radio transmitters. But unlike human-made systems, the fireflies achieve this without wires or algorithms, relying instead on eons of honed instinct. A single beetle's flash might deviate by milliseconds, but the swarm's collective rhythm corrects it, much like a murmuration of starlings adjusts mid-flight to maintain fluid motion. Evolutionary biologists grapple with a chicken and egg paradox here. For synchrony to evolve, it must offer a survival advantage from the start. But partial coordination, say, a few fireflies flashing roughly in time, wouldn't necessarily help them stand out to mates or confuse predators. The system only becomes beneficial when it's already highly synchronized, creating a dazzling display that overwhelms predators with sensory overload or signals fitness to potential partners. So how did the first fireflies stumble into this behavior? Some theorists propose it began with simple competition males trying to outflash rivals, accidentally falling into sync through sheer density. Others suggest it emerged from predator evasion, where chaotic flashes might disorient bats, but these explanations struggle to account for the precision observed today. A few fireflies blinking randomly wouldn't create the hypnotic waves that draw tourists to places like Thailand's Mekong River. That requires a level of coordination that seems pre-programmed, as if the rhythm were written into their DNA. Even more mystifying is the fact that multiple firefly species achieve synchrony independently, from the mangrove-dwelling pteroptics to the Appalachian synchronous fireflies of North America. This implies the behavior isn't a fluke, but a repeatable solution, a biological algorithm that emerges under the right conditions. Yet the genetic and neural pathways enabling it remain poorly understood. How do genes code for a behavior that only manifests in crowds? How does natural selection favor a trait that requires mass participation to function? The firefly's light show isn't just a wonder of nature, it's a living puzzle, illuminating the blurred line between individual instinct and collective intelligence. Whether you see it as evolution's improvisation or a whisper of deeper biological laws, one thing's certain: in the darkness of those mangroves, thousands of tiny lanterns are writing a story science is still learning to read. The resurrection plant doesn't just endure drought, it cheats death itself. Selaginella lepidophylla, a humble moss-like fern from the Chihuahuan Desert, can lose nearly 95% of its water content, shriveling into a brittle brown tumbleweed, only to spring back to vibrant green life within minutes of encountering moisture. This isn't dormancy, it's biological necromancy, a defiance of everything we know about cellular survival. Most organisms wither and die when water vanishes, their proteins unravel, their membranes shatter, and their DNA fractures like dried parchment. But Selaginella, it laughs in the face of desiccation, deploying a molecular arsenal so ingenious, it rewrites the rules of life's limits. At the heart of this miracle are sugars, not ordinary table sugar, but trehalose, a molecule shaped like a microscopic shield. As the plant dries, trehalose floods its cells, forming a glass-like cocoon around proteins and membranes. Picture a ship preserved in amber, frozen in time yet perfectly intact. This sugar glass doesn't just passively protect, it replaces water, hydrogen bonding to biomolecules in eerily similar ways, maintaining their structure even as the cell's liquid vanishes. The result, DNA that doesn't snap, enzymes that don't denature, and membranes that don't rupture. When water returns, the trehalose dissolves gracefully, like a thawed guardian stepping aside to let life resume. But trehalose is only half the story. Enter dehydrins, specialized proteins that act like molecular shock absorbers. As dehydration stresses the cell, these proteins coil around sensitive structures, stabilizing them against mechanical forces that would otherwise tear them apart. Think of a demolition crew wrapping a historic building in protective blankets before blasting nearby, except here, the building is the cell's machinery, and the blasting is the crushing weight of drought. Together, trehalose and dehydrins form a fail-safe system, a biological pause button that halts metabolism without irreversible damage. Here's where evolutionary theory staggers. Selaginella's adaptations aren't just useful, they're over-engineered. Most desert plants merely conserve water or deepen roots. This plant, it prepares for total annihilation, as if anticipating apocalypses that rarely come. Such extreme tolerance seems gratuitous in a world where droughts, however severe, eventually end. Why invest energy in mechanisms to survive years without water when mere months would suffice? It's like packing a spacesuit for a hike, brilliant but bafflingly excessive. Some scientists argue these traits are holdovers from ancient times when Earth's climate swung more violently. But that just pushes the mystery back. How did the first Selaginella stumble upon such a perfect toolkit? Trehalose production requires multiple enzymes working in concert, and dehydrins are useless without precise localization signals to guide them to crisis zones. Half-formed versions would offer no advantage. A cell with partial trehalose would still explode like a dried grape. The system only works when fully operational, a paradox that leaves Darwinian gradualism gasping. Even more perplexing is the revival process. When water returns, the plant doesn't just rehydrate, it reanimates. Membranes reassemble, enzymes reactivate, and chromosomes repair themselves with precision that humbles modern medicine. Human cells deprived of water die screaming. Selaginella's cells emerge from their sugar-coated crypts as if waking from a nap. What does this mean for life's resilience? From Mars missions to crop engineering, scientists are scrambling to harness these secrets, but perhaps the real lesson lies deeper. In a universe where survival hinges on adaptability, the resurrection plant whispers a radical truth. Life isn't just persistent, it's prepared. Whether by evolutionary foresight or freakish luck, this unassuming fern holds keys to immortality we're only beginning to grasp. The honeybee's waggle dance isn't just a quirky behavior, it's a linguistic breakthrough unfolding on the hive's dance floor. Imagine a scout bee returning from a field of lavender, her body buzzing with urgency. She doesn't just lead her sisters to the treasure, she tells them where to go, translating the language of sunlight and geography into a choreographed cipher of movement. The dance, a figure-8 pattern punctuated by vigorous waggles, encodes precise GPS coordinates. The angle of her body relative to the sun reveals direction. The duration of her waggle run spells out distance, and the intensity of her vibrations hints at the nectar's quality. It's not just a signal, it's a symbolic system so nuanced it rivals human telegraphy. What makes this communication truly staggering is its abstraction. The dance doesn't physically point to the flowers, it represents them, a cognitive leap once thought exclusive to humans. For the hive to act on this intelligence, every bee must share an innate understanding of the code. A forager interpreting the dance must mentally convert waggles into flight vectors, adjusting for the sun's movement since the scout's return. This isn't instinctive mimicry, it's a shared computational framework, as if every bee's brain runs the same navigation software. Evolutionary biologists face a conundrum here. For such a system to emerge, each component, sender, receiver, and the dance itself must evolve in lockstep. A bee that waggles randomly gains no advantage. One that deciphers non-existent dances wastes energy. The entire communication network only becomes beneficial when fully operational, raising the specter of irreducible complexity. Some theorists propose simpler precursors, like pheromone trails or basic vibration signals, but the leap from chemical cues to symbolic dance is colossal, like transitioning from smoke signals to Morse code. Even more perplexing is the dance's adaptability. On vertical hive walls, bees transpose the sun's angle to gravity, with upward waggles indicating flight toward the sun. In darkness, they rely on magnetic fields. This flexibility suggests not just hardwired behavior, but a grammar, rules that can be contextually adjusted. Human language operates similarly, with syntax adapting to environment or culture. The parallels are uncanny, begging the question, how much of what we call language is uniquely human, and how much is woven into life's broader tapestry? Critically, the waggle dance isn't foolproof. Miscommunication happens. Bees sometimes misinterpret angles or distances. Yet these errors reveal something profound. The system isn't robotic. It's a living language shaped by noise and nuance, where meaning emerges from collective agreement rather than rigid programming. The origins of this behavior remain shrouded in mystery. Did it begin with random movements that gradually standardized? Or was there a sudden cognitive leap, a behavioral mutation so transformative it rewired hive sociology? Neither explanation fully satisfies. The dance's precision demands coordination beyond chance, while its complexity resists simplistic, just so stories. In the end, the waggle dance doesn't just challenge our understanding of insect intelligence, it blurs the line between computation and communication. Whether you see it as evolution's improvisation or a glimpse of deeper biological logic, one thing's clear. In the hive's dim light, a tiny dancer is rewriting the rules of what language can be. From molecular machines to migratory mysteries, these 22 biological wonders don't just stretch evolutionary theory, they snap it like a twig under the weight of their own sophistication. Each one stands as a testament to life's boundless ingenuity, challenging the notion that random mutations and natural selection alone could engineer such precision. Take the flagellum's irreducibly complex motor, or the firefly's perfectly efficient cold light systems so finely tuned, they seem to defy the very logic of step-by-step assembly. Even Darwin himself acknowledged the limits of his theory when faced with the eye's perfection. Yet today's discoveries reveal puzzles far more profound than he ever imagined. What's striking isn't just the complexity, but the consistency. Across species and ecosystems, from the depths of the ocean to the canopy of rainforests, life operates with a fluency that suggests more than mere accident. The genetic code's universal optimization, the octopus's shape-shifting camouflage, the Arctic tern's celestial navigation, these aren't isolated quirks. They're threads in a tapestry of engineering so advanced, it humbles our greatest technologies. Science has unraveled countless mysteries, yet these marvels remain on the frontier, resisting easy explanations. Some argue these gaps are just temporary places where future research will eventually fill in the blanks. Others see them as signposts, pointing toward a paradigm shift in how we understand life's origins. Whether you view them as challenges to be solved or clues to a deeper truth, one thing's undeniable: they compel us to rethink the boundaries of chance and design. The conversation isn't about dismissing evolution, but about grappling with its unanswered questions and staying open to the possibility that nature's blueprints might hold secrets we've only begun to glimpse. So what do you think? Are these wonders the products of unguided tinkering, or do they hint at something more intentional? Drop your thoughts in the comments below. We'd love to hear your take. And if you're hungry for more deep dives into life's most astonishing mysteries, hit that subscribe button. The journey to the edge of understanding is just beginning.